Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, how are you today? It's a biology studies video that involves meiosis. Happy watching! Let me introduce myself. My name is Aidah Faiziah Permana. I am student Biology Education Class 3B from Universitas Muhammadiyah, Professor Dr. HAMKA. Before start to material, I want to ask you, do you know meiosis? What is meiosis? Definition of meiosis. Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. This cell is our sex cell, sperm in males, egg in females. During meiosis, one cell divides twice to form four daughter cells. These four daughter cells only have half the number of chromosomes of the parent cell. They are haploid. Meiosis product our sex cell or gamete, egg in female and sperm in male. Meiosis can be divided into 9 stages. These are divided between of the first time the cell divides meiosis 1 and the second time it divides in meiosis 2. History of meiosis. Meiosis was discovered and described for the first time in the Origins age in 1876 by the German biologist Oskar Hartwig. It was described again in 1883 at the level of chromosome by the Belgian zoologist Edward van Benedict in Ascaris roundworm egg. The significance of meiosis for reproduction and inheritance, however, was described only in 1890 by German biologist August Weismann, who notes that two-cell division was necessary to transform one diploid cell into four haploid cells if the number of chromosomes had to be maintained. In 1911, the American genetic Thomas Hunt Morgan detected crossover in meiosis in the fruit fly Drosophila melanogaster, which helped to establish that genetic traits are transmitted on chromosome. The term meiosis is derived from the Greek word meaning listening. It was introduced to biology by G.B. Farmer and G.E. East in 1905 using the idiosyncratic rendering meiosis. We propose to apply the term meiosis of meiotic phase to cover the whole series of nuclear changes included in the two divisions that were designated as heterotroph and homotype by Fleming. The spelling was changed to meiosis by Cornell in 1905 and by Pantel and the Senenti in 1906. To follow the usual convention for translating Greek. This is a picture of process of meiosis 1 and meiosis 2. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 1 is by far the longest phase of meiosis, lasting 13 out of 14 days in mice. During prophase 1, homologous maternal and paternal chromosomes pair synapse and exchange genetic information by homologous recombination, forming at least one crossover per chromosome. This crossover becomes visible as cosmata. This process facilitates stable pairing between homologous chromosomes and hence enable accurate segregation of the chromosome at first meiotic division. The paired and replicate chromosome are called bivalent 2 chromosome or tetrad 4 chromatin. Which one chromosome coming from each parent? Prophase 1 is divided into a series subtypes which are named according to the appearance of chromosome. Metaphase 1. Homologous pairs move together along the metaphase plate as kinetochore microtubule from both spindle form and attach to their respective kinetochore. The paired homologous chromosome link along an equatorial plane that bisects the spindle. Due to continuous counterbalancing, force extended on the bivalent by the microtubules emanating from the two kinetochores of homologous chromosome. This attachment is referred to as a bipolar attachment. The physical basis of independent assessment of chromosome is the random orientation of each bivalent along the metaphase plate, with respect to the orientation of the other bivalent along the same equatorial line. The protein complex cohesin holds sister chromatic together from the time of their replication until anaphase. In meiosis, establishing tension ordinarily requires at least one crossover per chromosome pair in addition to cohesion between sister chromatic. See chromosome segregation. Anaphase 1. Kinetochore microtubule sorted following homologous chromosome, which consists of a pair of sister chromatids to opposite poles. Non-kinetochore microtubules length, pushing the centrosome farther apart. The cell elongates in preparation for division down the center. Like in mitosis, only the cohesin from the chromosome arms is degraded, while the cohesin surrounding the centrosome remains protected by a protein named sugosin, Japanese from guardian spirit, which prevents the sister chromatic from separating. This allows the sister chromatic to remain together while homologous is segregated. Telophase 1. The first meiotic division effectively ends when chromosomes arrive the poles. Each daughter cell now has half the number chromosome, but each chromosome consists of the pair chromatid. The microtubules that make up the spindle network disappear, and a new nuclear membrane surrounds each haploid set. The chromosome uncoils back into chromatin. Cytokinesis. The pinching of the cell membrane in animal cell or the formation of the cell wall in plant cell, occurs completing the creation of two daughter cells. However, cytokinesis does not fully complete, resulting in cytoplasmic breaks, which enable the cytoplasm to be served between daughter cells until the end of meiosis II. Sister chromatic remain attached during telophase I. Cell may enter a period of rest known as interkinesis or interphase II. No DNA replication occurred during this stage. In prophase II, we see the disappearance of the nucleoli and the nucleal efflux again, as well as the shortening and thickening of the chromatid. Centrosome move the polar regions and arrange spindle fiber for the second meiotic division. In metaphase II, the centromere contains two kinetochore that attach to spindle fiber from the centrosome at opposite poles. The new equatorial metaphase plate is rotated by 90 degrees when compared to meiosis I perpendicular to the previous plate. This followed by anaphase II, in which the remaining centromeric cohesin, not protected by zygosine anymore, is cleaved, allowing the sister chromatic to segregate. The sister chromatic, by convention, are now called the sister chromosome as they move toward opposing poles. The process ends with telophase II, which is similar to telophase I and is marked by decondensation and lengthening of the chromosome and the disassembly of the spindle. Nuclear efflux, reform, and cleavage or cell plate formation eventually product total of 4 daughter cells, each with a haploid cell of the chromosome. And yeah, this is our final. Meiosis is now complete and ends up with 4 new daughter cells. Thank you for attention. Thank you for watching my video. Wassalamualaikum warahmatullahi wabarakatuh. See you.